Hello, everyone. Thank you for the introduction. So I would like to show you today what we do in my team, in my lab, the CSI lab at Novartis. I know it's a bit of a funny title, but I wanted to really show how much of the work we do really has of the detective quality in the day-to-day business. So the agenda for today for this presentation is to present what is the CSI and its role within the Novartis technical R&D division, how we do impurity identification at Novartis for new chemical entities, and then go through the structure elucidation and structure verification workflows in the team in technical R&D, and also how we work to understand drug degradation at Novartis. The first topic, well, this is not exactly what we do. So we are not routinely looking into debris or things that are on your shoes, but we do a lot of structure investigation in our team, and actually CSI stands for chemical structure investigation. We are responsible for structure elucidation, impurity investigation for the entire new chemical entities division actually in Novartis, both for compounds in development, but also for already commercially available products. So our mission is to be a one-stop shop within Novartis TRD and technical operations. We apply state-of-the-art techniques for impurity and degradant isolation and identification for small molecule and mixed modalities projects. We also perform forced degradation studies with the aim of understanding degradation pathways of our APIs and our drug products. We support project teams to gain better understanding of the impurity profile of DS and DP, and in case of troubleshooting throughout development and post-commercialization by providing fast, reliable, and high-quality data. We also manage impurity elucidation data across projects and through the years, and you can see on the right a little bit the different aspects where we are involved. And I'm going to go through the details of each application in the next slides. So as you can see, the main activities of the team are related to isolation of bi and degradation products. Normally, we isolate compounds in the 0.5 to 5 milligram scale for structure elucidation purposes. We sometimes go as low as 0.03 or 0.05% impurities in the main component, and our focus is primarily, of course, on actual impurities in drug substance, but also in intermediates and starting materials of synthesis, in degradants and excipient compatibility challenges in the drug product. And we try to provide to the project teams a full elucidation, so not only LC-MS, but also to the NMR elucidation to be able to give them that certainty in the structure elucidation that can then be submitted in the regulatory dossier. We perform forced degradation studies, both for drug substance and drug product. And we also, as part of these studies, isolate and perform structure elucidation of the major degradants. And together with the project team, submit for mutagenicity assessment. We also are involved in the assessment of the analytical method suitability, meaning if the method is stability indicating, and also in the definition of the degradation pathways. Of course, as I was saying before, we don't only work for clinical projects, but also for commercial projects whenever there is an out of specification result or troubleshooting for these products. We also provide brand maximization support, so that's a profiling versus infringers, for example, work. And of course, in case the project team has difficulties in synthesizing impurity, we also perform semi-prep, so small scale isolation of in 100 milligram to 1 gram scale, for example, for an Ames test performance. In addition to that, we perform also routine NMR and MS support for project team for structure verification, as well as more complex verification and elucidation of compounds that have been synthesized in the labs. And normally we are involved in around 400 elucidations per year and 700 structure verifications. We also work in the impurity data management. We have our own database that is based on instant JChem and are onboarding now our data and our compounds into Luminata, which is an ACDLabs software that you might be aware of. So this is a little bit how the different activities in the pie, you can see the different activities make up our overall capacity and portfolio. The technologies, I briefly showed those in the previous slides, but more specifically, we have 2D LC-SPE QTOF, we have an Orbitrap high-resolution MS, we have access to Orbitrap GC high-resolution MS, several semi-prep HPLCs. We are also introducing the Chromacon Cube prep LC, which is a biocompatible twin column prep LC for the purification, isolation of impurities, mainly in peptides and oligonucleotides projects, and of course several UHPLC and HPLC instruments, as well as access to several NMR instruments together with our research colleagues up to 600 megahertz. Going a little bit on how we do the work related to impurity identification at Novartis, I'm going to show you a little bit the different ways that we tackle that. In early phase, our main focus is on the API and on the drug product degradants that we observed in ASAP studies, so in accelerated predictive stability studies. We perform routine identification of process impurities in earlier synthesis steps only later. In the early phase business, we mainly do tentative LC-MS for these earlier synthetic steps. As we move into late phase development, references become available, allowing full structure elucidation also for the impurities present in the starting materials and in the intermediate of synthesis. Normally, these references are obtained through isolation in batches or samples where the impurity is present, and this is done then in the CSI. And in some cases, if the structure is also easy to synthesize, we have it synthesized by the project team or at an external CRO, and then we perform the elucidation of that reference. In addition, throughout development, we are also involved in different structure verification activities. For example, if we have a resynthesis of a reference that we need to assess that this is again the right compound. So beyond the elucidation, several verification activities are ongoing in parallel. This is a little bit our early phase workflow for impurity in the API, impurity identification in the API. So you can see a little bit how much it takes. This is done mainly using LC-MS, at least for the ones where we feel we have a quite good confidence in the MS data. And for the other, where we feel either the LC-MS data are not sufficient to provide a good match in terms of structure, or we see a potential of having an alerting structure in terms of mutagenicity, then we would go for a full confirmation. So you can see that we start normally from a data collection. So we perform an LC-MS using mainly a 2D UHPLC system. Then together with the chemist, there is a structure proposal that is done based on the process knowledge and the impurity molecular mass. We do then a match by MSMS. We use mainly here Unify software from Waters, where compare the proposed structure with the fragmentation observed experimentally for the impurity at hand, and then we do an evaluation by direct to see if there is a potential mutagenicity alert in silico. In case indeed we do observe there is either an in silico alert, or the structure cannot be, let's say, with the Unify matching sufficiently clarified, we would go with isolation and enrichment. Here we have different approaches. I would say that in the CSI team, we mainly go for semi-prep LC, although in some cases where that has not been possible, we've also used online SP. And to that, we then would add the NMR, normally to the NMR and MSMS on the isolated impurity for the structure confirmation. So as I mentioned already, for the early phase projects, the main focus is on those steps 1 to 3, and I think this is really very, so say, diluted timelines where we really feel that, to be on the safe side, we would be able to do this within 2 weeks, but it really depends, of course, on the number of impurities. If it's only 1 or 2, this might run really within very few days. And for the activities 1 to 5, I would say that for most projects, this is for more later phase, or when we don't have a confirmed structure. This for most projects takes actually a week, but sometimes we would actually need more time if the very low levels of impurities are present, like below 0.05 in the matrix. So this is a little bit what I was saying. So it really depends on the complexity, but I would say that overall, if we are going to do it for every project, this is the timeline. So we start planning 4 weeks in advance, and then normally 1 week for the structure proposing and matching. And if you need to more, it can take a bit more. It depends a lot, of course, on the NMR capabilities. We have very good access. We have access to very good NMRs, and this allows us actually normally to have most of the challenging structure confirmation activities completed within a couple of weeks. So this is really the full structure elucidation workflow. It's slightly different than what we would do in early phase. So it's really targeted to looking at that specific peak that we are interested to understand what this impurity can be. So we start from the unknown chemical structure and we perform directly the PrEP LC. So we do the PrEP LC method development, and then we isolate the peak. We lyophilize it and perform MS both before and after lyophilization to ensure also that there is stability after the lyophilization. Perform then 2D NMR and high resolution MS, and using the two inputs of both the mass spectrometry and the NMR together, we would end up with the final proposal of a confirmed, hopefully, structure that we would then have in ELN, share with the project team, and update in our impurity database accordingly. As I said, we go very low. We go even below 0.05, especially for commercial projects. And normally we say that these results are fully elucidated structures that can be then submitted for in silico, for mutagenicity assessment, and included in regulatory submissions. So in that perspective, we do this work mainly for the late phase activities. As I mentioned, though, we also have some automated structure verification activities that are embedded into our analytical data cycle. This is a project that we are working on together with our research colleagues. So there is a joint interest here to have data from both research and development, where we more or less have simple molecule below 800 Dalton that we have maybe even an idea of what the structure can be. So we are using an automated structure verification tool that is provided to us by ACDLabs. The idea behind is that directly the software is able to pull the structure from either the lab notebook or we have a database for a platform for actually submitting requests, and directly a 1D NMR a proton 1D NMR is triggered once the analysis is completed and the spectrum is analyzed, and based on that result, the experiments, the parameters of the experiment, the next experiment, are actually customized and sent directly to the spectrometer. Once the results are available, they are exported to the ASV and gene, and the result, which is hopefully a structure, proposal, a confirmed structure, is then directly reported to the chemist, to a database, or to ELN based on our needs. The reason that we decided to introduce ASV mainly for structure verification purposes also in development is that we see it as a time saver actually for our teams, and especially for the CSI, since our major focus is on structure elucidation. We want to work as little as possible, so to say, on verification, meaning on those cases where we feel that we know already what the structure is, or there are already data in the database about this structure that was perhaps already analyzed in the past. So we have different types of results. We can have a complete verification done by ASV, and then actually the associate, the expert's time on reviewing the result is normally 1 to 2 minutes, which is more or less 1/10 of what the manual analysis would take, which is around 20 minutes we consider for a structure verification. There are sometimes we need to do a minor manual correction required, and that is around normally up to 5 minutes maximum work from the expert. And then we have also some case where the ASV failed, meaning that we either didn't get a result at all, or not everything was processed. Normally, at least the big picking that was done as part of the initial ASV result is still usable, so we still see a reduction in the analysis time from the expert. You see here are actually that we've been working already for 2.5 years with this, also in development. In this case, we were using over 100 megahertz in one of our labs, and you can see that with this assistant open access, meaning that the associates, the chemists, were directly going, submitting their NMR tube to the analysis, and then they were getting a request. At the background, there is the ASV running and an expert that is checking that this all is working correctly. So we got around 45% of complete verification with ASV only, 27 where we had a minor problem and we had to intervene, and 28% where we had a failed verification. We are now around 60 to 65% success rate at the moment after 2 years. So we've really seen an increase in the green results, and the software really seems to be learning, of course, by as we feed more data into it. And I wanted to spend the last part of the discussion on how can we actually predict degradation and how we work in relationship to degradation understanding in the CSI and in Novartis. So we mainly have 2 types of studies. We have studies that are focusing on understanding the degradation profile, so the post-degradation studies. And these are the ones mainly then also performed in my team in the CSI. But we also have, of course, the ACDLabs studies, which are mainly aimed at predicting the degradation rate. These are performed between the analytical department and the pharmaceutical department with the aim of understanding the kinetics of the degradation and of the formation of the specific degradants and the dependence of the rate on temperature and humidity. So I just wanted to in relation to that, show a little bit the different identification activities that take place during the development roadmap at Novartis. So pre-DC, so before we actually declare a compound as a drug candidate to go into first-in-human trials, we perform mainly tentative ID, mainly low resolution. Some deeper investigation are performed for parenteral and topical projects, as there we need to lock the formulation, especially for the topicals, quite early. And then once we are still in the before first-in-humans, but we have the first, so to say, GMP manufacturing, we perform, and the first formulation, we perform the early phase first degradation and the asset for the clinical supply form formulation. And here we have tentative ID that is routinely taking place, both as part of the method development and as part of the ASAP, and we are able to propose already at that level the first degradation pathways and understand the major liabilities of our compound. As we transition into late phase, so after proof of concept, we are focusing on more in-depth and full identification type of studies. Here we have the degradation pathway studies. That was the one that I mentioned briefly occurring in the CSI team. And then we have also late phase ASAP and late phase FORSTEC that are mainly related to regulatory requirements or to late phase validation activities for the stability studies. All these identification activities in the late development space are aiming to have as much as possible a confirmed structure that we can submit for an in silico assessment, and of course include in our regulatory submissions. I have to say at Novartis we're very strict, so to say, with that, and we really place great value in having fully elucidated structures already around POC for the major impurities and degradants. So you can see a little bit for the degradation pathway study, which are the different tools that we use. We start with the first discussion with the project team about the study, and we start with the protocol preparation and the initial in silico prediction, where we used Zenith as a tool to help us understand what are the potential liabilities and type of pathways that can occur. We then perform the study and analyze the results, and we identify the major degradants. Normally this is done following isolation, NMR, and high-resolution MS. For the MS, we often use the Waters Unifier system, or of course Excalibur if we're working with the Orbitrap. And for the NMR part, we use Spectrus and introduce this data then also in Luminata. We update the team, of course, and the impurity database as just mentioned, and then perform the in silico screening of the major confirmed degradants for mutagenicity with Derek, Sarah, normally, and an expert assessment. And we also then at the end, for the report, prepare a degradation pathway proposal together with a chemist. And here we also, besides literature, of course, we also use the Zenith tool. This is a little bit our idea of how we would then manage all this input, be it from the degradants or for the impurity identification activities. As you see, there are different types of data that we are interested in managing in terms of impurities, be it from the process development, from the QBD experiments, the carryover of impurities, the project, the campaign data, the GMP data, that is, the reference standards, the impurity identification of the CSI, but also anything that the chemists will do on an open access format in our research department, or the degradation pathways that are occurring at different parts of the organization. So these data go in different, so say, platforms, but we are using Luminata a bit as the common point where we would actually bring all this impurity information together, be it from ELN, be it from ASV, and this is actually where the ASV also comes together in this picture. And out of these two, so out of ELN and Luminata, we would actually feed in the regulatory documents and in Spotfire. I think maybe just a couple of words. You might have seen this from Luminata, from the ACDLabs, but I just wanted to show you what is the main use that we have in Novartis out of Luminata. So we mainly use it to have a better understanding and an overview of our processes. The idea is that we would depict the process map, the process scheme, with all the impurities, the impurity map attached to it. And you can see then here at the bottom of the page that for every then impurity or for every compound, we have full information regarding that chemical entity, like the structure, the Bruder formula, and the the molecular weight, and we also know where, at which stage, and in which batches that impurity was observed. And on the right side, you can see that we get then all the data, all the analytical data, like the reference analytical data, the HPLC of that specific batch, or the NMR of that specific compound. So we're able to capture at the same time the information regarding the reference, the specific batch, also though, and that is most important, how this connects to the different synthetic steps and where this was observed. One other aspect that we are looking into in Luminata is the stability data management. We have just started looking into it, and we see this as quite interesting, especially for our early phase activities in the field of degradation understanding, as this we feel can support the workflow where we would introduce the Zenith, for example, data and get the support for the tentative ID by LCMS. It's actually very nice because you can directly introduce in the software the Zenith file and create a degradant map and associate directly the LCMS data to it. For the late phase so far, since as I said, we operate on a full structure identification basis. We are mainly working with a compound database within Luminata, meaning that we are bringing all the information and then building a map on our own. But we will be looking if there's a potential of this stability data management within Luminata also for the late phase activities. And I think just one final slide on the Luminata. The whole idea behind it is that we would be able, in 3 simple steps, to have a full information, so to say, on the compound. So we have the separate components that are introduced in the software. We have the possibility, based on the mass, then to identify those components, and of course later to store, to share. And this is especially important in the area of the degradation, as it is very, we're working there with many different departments, and it's crucial, the collaboration and the data exchange. You can see here that I think for us the most important part for the compound database is how we will be able to share those data between the formulation, the analytical, and the chemistry department. And we feel that this is actually where we can really make a difference with the tool. This is just a map showing where we are actually planning to use or already using Luminata as part of our data management. So you can see that we start from here at the bottom, the raw materials or the non-contributing raw materials, and the API starting materials and the intermediates, so the process data, and then at the end, the drug substance. And then on this horizontal axis, you see the early supplies, the late supplies, and the launch. And as you can see that while in the early phase, we would focus mainly on the reference standards for the API starting materials, and only for the DS would we perform the identification activities of the impurities. As we move into late supplies, this identification then covers all other activities related to the synthesis and process finalization as well. So be it carryover of impurities, be it actual and potential degradants of the DS, but also, of course, reference standard establishment and updates, actual impurities in starting materials and intermediates, as well as all the activities we perform on a transactional basis for our compounds. The idea is that we have something that is ready to be shared with our colleagues in the technical operations, and if needed, if other information are coming on certain projects, also from commercial, these information then can be also fed back into Luminata so that we have a single repository for all our data. So that's a bit how we handle the data within the Novartis CSI, and also I would say the Novartis TRD, and the different techniques that we use to identify impurities and manage those data. Acknowledgement goes, of course, to all the team, the CSI team, but also Adrian Clark and the colleagues from our research department, and of course to the ACDLabs, who has been providing some wonderful support through these years to implement Illuminata. And I'd like to thank them also for the opportunity to present here today. And I'm like to open then the scene for your questions. Thank you.